Okay, aloha everybody, and welcome to our next chat here on our virtual seminar series, our virtual guest lecture series at BYU Hawaii. Today, we're very excited to have Dr. Jeff Stogsdill with us. Welcome, Jeff.
Hi, welcome. Thanks for letting me join in today.
Yeah, thanks for coming. So Jeff's currently quarantining in Virginia, but he is usually based, and hopefully will be returning shortly to Harvard University. So Dr. Stogsdill is currently a postdoctoral fellow in the Department of Stem Cell and Regenerative Biology at Harvard University. He started his education at Brigham Young University in Provo, where he did Bachelor's of Science in Physiology and Developmental Biology, and he finished that in 2010. He then continued on to do a master's also from BYU in the Department of Physiology and Developmental Biology with Dr. Paul Reynolds, and he finished that in 2012. And then he went on to do a PhD in the Department of Cell Biology at Duke University in North Carolina, and he finished that in 2017. And that's where we met. I was doing a postdoc in North Carolina at the time. And then he went on, starting in January of 2018, he moved up to Harvard University for his postdoctoral fellowship. So we're really excited to have Jeff here today. He's going to talk a little bit about his research, which focuses on trying to understand the molecular communication between neurons and non-neuronal glial cells that are found within the developing central nervous system. So this is really exciting research that has a lot of really interesting implications for different nervous system disorders. And we're excited to hear more about that. Before we do that, though, I just wanted to talk with Jeff a little bit about sort of his background and his path and kind of how he got to where he is today. He's in a great postdoctoral position at a university that I think most people have heard of. So we want to hear a little bit more about how he got there. One thing that my students are often surprised by is how specific people end up getting, how specific faculty members in particular end up becoming in their field. And Jeff, you're currently researching a very specific part of the nervous system. How did you tell us maybe a little bit about how you got interested in neuroscience? Is this something that's been of interest to you for many years? How did you get into to this field of neuroscience?
Yeah, that's a great question. So I have to be completely honest. I am a neuroscientist that has never once taken a neuroscience course in my life. I
Congratulations.
yeah, in high school, I found biology extremely fascinating. I thought I wanted to go to med school, so I went to BYU for undergrad with that intention. I fell in love with a few developmental biology courses that I took, learning about the signaling that occurs during development, how you go essentially from one cell to a whole organism. That was, I found that extremely fascinating. It just so happened that when I got to grad school, that a lot of the developmental biology labs were focused on neural development. And so I did a couple rotations in developmental biology labs that were studying different aspects of CNS development or regeneration, and I ended up choosing one of those labs and have loved it ever since. I'm still not a huge fan of the electrophysiology and the nitty gritty of neuroscience, but the neural development is my thing.
Cool. So you mentioned that sort of going at you had this fascination with biology, and you went into your university studies anticipating perhaps that you would end up in medical school. I think this is very common. What turned you from that medical path to sort of more of a research focused path in academia?
Yeah, so I just found I really loved research. The ability to be in lab, to do something that no one has ever done before, or ask a question that no one's asked before, and to get an answer really intrigued me.
Yeah.
The MCAT also was definitely a portion of that.
Deterrence.
A little bit of a deterrent. I ended up doing okay, and that's what got me into some of the schools that I got into. But yeah, I think first and foremost.
So did you actually apply to med school?
I didn't apply to the med school, but
Okay,
I used my MCAT score, and that helped me get
gotcha,
into, for sure, the master's program,
gotcha.
and it didn't also hurt for grad school. Um, but yeah, so I would say first and foremost it was the love for research, and then I also had wonderful mentors along the way,
Yeah,
particularly in as an undergrad and then as a master's student. My my mentors were phenomenal in helping me shape what my interests were and finding my little niche in science.
yeah, great. And I think there's super important lesson there, especially for undergraduates, is to get involved as much as possible with mentors of with diverse interests to try to figure out what those career paths are that are out there that are possible for students, because oftentimes that view is limited. I kind of operated in a similar way when I was much younger. I thought, I loved animals. I wanted to work with animals. I'm going to like work in a zoo, right? I mean, that's like what I thought I had to do if I was going to work with animals. And I became exposed over time to, to researchers who were studying animals in lots of different ways all over the world. And they were all doing that in sort of a research in an academic research context. And so I think it's really important to point that out, and to also, I think as we listen to your research a little bit more, I hope that people will kind of see some of the connections between, things that you're doing, research that you're doing, and research that many other scientists that are doing around the world that are solving problems or contributing to the development of solutions that impact our everyday life. And many of those overlap with medicine. And I think I agree. I mean, I'm right there with you in terms of the fascination with that research process and that prospect of discovery, which is not always something that you get in sort of the day-to-day grind of a family physician. And that's great, and
Right.
that's it's something we need, and something that a lot of people love to do and are very dedicated to.
Yeah,
And I
one
think
thing I'd add, I mean, growing up, you would know of famous physicists, right? Stephen Hawking or,
sure,
Albert Einstein, but you never heard of famous biologists, or you've heard of like Jane Goodall as a famous,
sure,
scientist out in the field. But I think there were very few of those role models. And I think those are coming on board now a little bit more.
yeah.
And so you're exposed a little bit to those potential research career paths that maybe I didn't seek out as a kid, or I just wasn't exposed to.
That's my experience is very much the same. It wasn't until I, started interacting with people who were professors and who were studying the things that I was really fascinated by that I realized that was an option.
Yeah.
And I think it's particularly, important for, a lot of people who I meet that are, thinking about going into med school, for example, are doing so because they have, maybe some history of some family, of some illness in their family, and they're really interested in, sort of contributing to, taking care of people in those areas. But oftentimes a really exciting, research that's, contributing to a cure for many of those conditions happens in the lab, and it's not happening, necessarily in a hospital or in a doctor's office. And that's another reason I'm looking forward to hearing a little bit more about your research. So moving on a bit, you mentioned, we talked about how you did a master's program following your bachelor's degree, both at Brigham Young University. This is something that oftentimes I have students who are interested in pursuing graduate work who come to me and wonder, should I jump right into a PhD? Should I do a master's? What are some of the trade-offs there? Perhaps you can tell us a little bit about your experience doing a master's degree and kind of how that shaped your career trajectory from then on out.
Sure. Yeah. So the decision to go into the master's program was brought to me by my mentor at the time at BYU, who he had a position open in his lab. And he offered to me. He knew that I had recently been married, and I was kind of in that waiting period. My wife was still needing to finish undergrad herself. So I did have a little bit of a year and a half time period in which I could do this. it was also an opportunity for me to boost my resume, my CV, to help me get into grad school, and without question, that definitely occurred.
Sure. Yeah.
I was able to get 2 publications as a master's student, and I think that was hugely beneficial. Uh, I would say it's not critical by any means that you have to do a master's. In European countries, for instance, it's kind of lumped in with the either the undergrad or the PhD portion.
Right.
Here, I think you just need the experience, either as a master's student or as working in industry, or even just as an undergrad, having significant lab experience will help you get into the research programs, the PhD programs that you might be of interest.
Yeah, great. Did you also find maybe that it helped kind of hone your interests a little bit at that point?
Um, it definitely helped build kind of my scientific knowledge and base a little stronger. What it did actually for me was focus a little too narrow of what I thought I wanted to do. So I went into grad school with the full intention to continue kind of on that same path as I did as an undergrad and master's student, which was studying lung development. That obviously ended up not happening. So I had to broaden my horizons a bit once I got to grad school. But at least I was able to pick up on some new techniques as a master's student, understand kind of what life was like a little bit as
Sure.
a PhD student, which is totally different than undergrad.
Completely different. Yeah, I'm always students are always shocked when I give a little talk every year at our science career fair, and I kind of talk about grad school and how to get into grad school and what what a day in the life of a grad student is like. And people are often shocked that in the life science in particular, there's a lot of financial support for students. In many cases, you're not taking out loans and paying tuition and going into debt. You're actually earning a stipend, and then also just in terms of the absence of coursework in a lot of cases, especially after your initial couple of years, people are often times kind of surprised by that. You're not just in the books and studying for 5, 6, 7 years straight. It's very much research driven and
Right.
and inquiry driven, which I think is why a master's program is really helpful, especially if people haven't been exposed to research earlier on, or if they're in kind of one of those transitional periods, which is really helpful as well.
Right. The way that my mentors have told me about it is that as a PhD student, your goal is not to rewrite the textbook, but to actually just write it new.
Right.
So that's what the research is for, is to build upon what's already known.
Yeah, absolutely. Absolutely. Great. Well, I had one other question that popped into my head and then it popped out. Maybe it'll come back to me later. So perhaps now we can, just kind of turn the time over to you for a few minutes to share a little bit about your research. So for my students who are out there watching, Dr. Stogsdill is going to share a little bit about, kind of what, I know what I wanted to ask. Sorry, before we go into that.
Sure.
You're a postdoc now, okay? So you went into grad school, you take some courses, you do a lot of research on your own, you're working with a mentor and everything. Transitioning into this postdoctoral phase, what is that like? What's a day in the life like for a postdoctoral fellow at Harvard or at anywhere else?
So maybe I can start what life was like as a grad student first for
Sure.
me, and kind of that transition. So really, I was a lab junkie. I just, I enjoy being at the bench. So every day as a grad student, I'd show up at the lab, and every day was something new. Every afternoon, every morning was something totally new, doing a different experiment of some kind. And so I would be there, 10, 11 hours a day, just, playing with cells, playing with mouse brains, doing different kinds of experiments. It allowed me I did fairly well in grad school, got 2 really high publications, which was nice. And it offered me the opportunity to go to international conferences and to meet with heads of labs and world-renowned scientists, and that helped me land the position, the postdoc position I'm in now. I met my boss as a graduate student. She was giving a talk, and I approached her and had a great conversation with her. And a couple years later, I emailed her again, and she remembered who I was and immediately offered a spot in the lab, which is wonderful. And so now as a postdoc, I do almost the same like what I do as a grad student. I don't take any classes now, which is nice. But again, I'm still at the bench full time now, obviously not during this COVID period. I don't do anything at the bench. So it's a lot of reading and writing now, but I'm anxious to get back to the lab to be able to run experiments again.
Cool. Yeah, kind of your experience is really similar to mine in terms of, kind of daily life as a grad student, as a postdoc, and then also importantly, how I found my postdoctoral position. And I think that really highlights the importance of, putting yourself out there, especially at conferences at both an undergraduate level and when you're a graduate student, because those connections that you make are, critically important when it comes to landing the next stage of your career. And I had a really similar experience meeting my postdoctoral advisor. I knew who she was. I knew about her research. I approached her and talked to her. I had met her husband while we were hosting him for a seminar. And so I used that as kind of a entry point. And then that kind of opened up this dialogue, and we applied for a grant together and we got it. And that, and that sort of brought me to that point. And so I think those types of experiences are, incredibly valuable, and science in that way is very, very social and very much about connections and making those connections so that you can, link up with people who are doing what you're interested in doing and who have a place for you to do that and funding for you to continue to do what you want to do. And I think that's something that oftentimes...
That was probably one of the more surprising things to me about science. I always envision, you're at the bench, you're by yourself, you're, in a dark room. Yeah. But no, it is very social. It's very interactive, very collaborative. And that's how science gets done. So it's a lot of fun.
Yeah. So, I mean, we scientists, we're definitely nerds, and we definitely love being in the lab or in the field and everything. But yeah, when it comes down to it, being able to, I think a lot of the most successful scientists out there are also incredibly good networkers and are very good collaborators. And that really helps, sort of push science forward, building those diverse networks of collaborators. Cool. Okay. Well, now I got that question out, it came back to me. I'm glad we had that discussion. Let's turn the time over to you, and you can share your screen with us, and then tell us a little bit about your research and kind of the field within that your research sort of falls within.
Yeah, absolutely. All right, you can see the screen just fine?
Yep, looks great. Go ahead.
Okay. All right, so as we've kind of discussed, my background in cellular and molecular biology mixed with my interest in developmental biology has really kind of shaped my focus and trying to understand how the brain is put together, both at the cellular and molecular level. So what you're actually seeing here on this initial screen, the image on the left is a mouse brain that has been, a whole mouse brain that has been cleared. And each of those tiny little green dots is a cell called a microglia, which is an immune cell that exists within the brain. The image on the right is a cross-sectional view of that same brain. Again, the green are these microglial cells, these immune cells. But you can also see in there these red dots, which are indicating the presence of these specific subtypes of neurons. And so really what I'm interested in as a postdoc is understanding how these two major classes of cells, neurons and microglia, and all their subtypes interact to be able to form a healthy functional brain, and then ultimately understand what happens when those interactions go awry that lead to neurological disorder. There we go. All right, so this is the human brain. You probably don't think about it all that often, and I wish as a society we probably should, but really it is the source of cognition, and it allows us to do the incredible things that we're able to do each and every day. Be able to compose music, me not so much, or be able to, do complex math problems, create works of art, to use reasoning to be able to understand, our place in the universe, to understand the cosmos, and, metaphorically here now, understand the brain itself. And when it develops properly and when it is well taken care of, you can do these things. But so how is it built? So just to talk about some of the numbers, these are numbers that I think are completely staggering to me. So in the typical human adult brain, so anywhere from the age of 25 to 55, you have roughly 86 billion neurons. So these are, the prototypical cell that you think of in the central nervous system. They have, they send axons, and they receive signals and move signals back and forth neuron to neuron. So there's about 86 billion of these in the human adult brain. If you take all the cells in the brain, it's about 172 billion cells. So that's 172 with 9 zeros at the end of it. These include other cell types, which I'll talk about later in the talk. And then all the connections between those neurons, which are called synapses. These are specialized junctions, which allow transmission of the signal from one cell to the next. There are roughly 164 trillion of these. So all of these different neurons, these different cell types, all of these synapses have to be produced. They have to be produced not only at the right time, but at the right place, and connect with one another in the right manner and organization to be able to produce a healthy brain. And again, when this happens, human beings can do marvelous things. But many times this doesn't actually occur in the right time and place. And that's when brain disorders arise. And this can come from either altered development or brain degeneration or any number of things in between. So things like ADHD or schizophrenia, depression, Alzheimer's disease. And I put up here brain injuries to highlight the fact that if you have a brain injury through, say, a concussion or through an injury, the brain has very limited regeneration capacity. It's not like your skin, which will heal a wound. Once injured, the brain does not restore itself. So any of these disorders or these diseases arise are long lasting. And unfortunately, we have very few treatments. And the treatments that we do have to be able to cure these are not cures. They're simply a Band-Aid covering the symptoms. So we're in a serious need to be able to come up with permanent cures for these neurological disorders. For instance, I'm sure most of your classes at least knows of somebody who has Alzheimer's disease, which is devastating not only for the individual, but for the family as well. So we need to find cures and treatments for this. But how do we do this? We can't ethically study someone's brain by taking it out, right? So sheepishly in this cartoon, there are a few human brains available to be able to study this. So instead, we use model systems. And some of these examples are shown here on this slide. Things like the roundworm C. elegans in the upper right, the mouse, old world monkeys like the macaque are used. In our lab specifically, we're now turning to things called cerebral organoids, the picture down in the bottom right, which are of human origin. These are human cells grown in a dish in a 3D spheroid or organoid shape that produce all the cell types that a normal brain would produce. So we can actually study human cells of a brain-like state in a dish. But for my research, I actually use the mouse. It's very comparable to the human brain, just much smaller, has all the same cell types, and produces a lot of the same functions, and even a lot of the behavioral outputs we can actually study using the mouse. So using the mouse, for instance, we can take out the brain here, and in cross view, this is kind of what it looks like. As a postdoctoral student, I actually study a portion of the brain called the cortex, which is that outer layer there. And I use it for a number of reasons, but it's a good model system because it has all the cell types that we're interested in studying. It's an area of the brain that is highly important for higher order cognitions and emotions, and for larger thought processes. And it's a hub for gathering and all the sensory information that we experience for a generalizable output. So the cortex is a layered structure. So it has 6 defined layers, and those layers are defined by these cell types called excitatory neurons. These are the GO neurons. And these neurons are not just, it's not just one type. There are many, many, many types of excitatory neurons. And we're interested in my lab to figure out how these different cell types arise and how they interact with other cells, like the inhibitory neurons. These are a different kind of neuron that put a stop to all the go signals within the brain. You have to have the proper balance of excitatory and inhibitory neurons to have a fully functional brain. And lastly, this other class of cells called glia. These are non-neuronal cells. They are found at equal numbers as the neurons in the brain. And I'm interested, as a graduate student, I studied astrocytes. And now as a postdoctoral fellow, I study microglia, which are the immune cells of the brain. And I'm interested in understanding how these microglia interact with the different subtypes of neurons. So these different main classes of cells, we can kind of throw them into different functional buckets. So neurons, for instance, as you're probably aware of, act as like the circuits of the brain. They come in a go form and a stop form, the excitatory and the inhibitory, respectively. I study these microglia, which are these immune cells which protect the brain from pathogens and from viruses, but they also help sculpt the neural circuitry. So they eliminate neurons that are not supposed to be there, or they eliminate synapses that are not needed anymore. And in healthy conditions, this happens all the time. However, in disease states, these microglia can go awry and start really chewing up the brain, which can cause major issues. And we'll talk about some of those later. So in general, I study the two of the major body systems in the body. Sorry, that's my son playing with a truck. So I study the nervous system, which are the neurons, and the immune system. Your immune system, you generally think of all the antibodies and the B cells and the T cells that are floating in your blood. But these the immune system actually has a huge part in your brain, which again are these microglia. So as a postdoc, I'm really fascinated by some of these major questions here. So what are the molecular signals that promote neuron and microglia development? How do microglia regulate specific neural circuits, and what effect does that have on your behavior and the way you think and the way that you form memories? What are the neuroimmune interactions that are disrupted or hijacked during neurological disorders? We're learning a lot about this now, especially in the past 5 years, that the immune system plays a huge role in these neurological disorders. And because of this large role, we're now keenly aware and looking for opportunities in where we can intervene to be able to cure some of these neurological disorders. Can we fix the microglia, or the immune system in the brain, to be able to restore cognitive function? So these are some of the big questions that we're trying to address in the neuroscience field in general, as well as in my own personal research. So just to kind of back up a little bit. So these cells have these different functions because of genes. We have tens of thousands of different genes that are encoded by our DNA. And these genes do different functions. So in a very highly simplistic form, you can see here that, for instance, gene 1 for this neuron controls its electrophysiological properties. Gene 5 controls its cellular morphology or its shape. Gene 4, for instance, guides its axon targeting, where it's going to signal and where it's going to connect throughout the brain. In reality, you have many thousands of genes that work together to be able to form these different functions. So how do we make sense of this? So in the lab I'm in, we use genetic sequencing to be able to read what the cell is doing, to read what it is. So we use new sequencing technology tools, like single cell RNA sequencing, where we can take a soup of cells, put each cell into a little droplet, and then read all of the genetic material within that cell. And we specifically use RNA as a readout. And that RNA can then tell us not only what that cell is. You can see here in this plot that all the different colors are of equal cell type. So all the reds are excitatory neurons from the upper layers of the cortex, whereas the blue cells are the inhibitory neurons. So we can read what these individual cells are doing. We can read what they are, but we can also tell what they're doing based upon their RNA pool that they're expressing. And this becomes really powerful, because then what we can do is we can combine and compare two different scenarios. For instance, we can compare a normal mouse with a diseased mouse. So we have mouse models for Alzheimer's disease, for example, or a mouse model in which the specific motor neurons that are damaged during Parkinson's disease, those are gone, for instance. So we can compare a normal versus a diseased mouse and compare the RNA transcriptomes on a cell-by-cell basis to understand what's going on in those cells. Or alternatively, we can compare different brain regions and figure out what's different between a cortex versus the striatum, which are two brain regions of interest in the lab. So then we can, by doing this, piece by piece, be able to understand how the brain is built and how it goes wrong in disease. So as a postdoc, really what I'm trying to focus on right now is understand and be able to construct molecular networks to infer cell-cell interactions. So what I mean by that is, as I've introduced, there are a number of different subtypes of neurons in the brain, which are labeled here in gray, for example. And I'm interested in the microglia and the signaling that occurs back and forth between those two major cell types. So these secreted ligands in green that are secreted from the neuron and signal the microglia, or vice versa, as well as cell surface receptors and ligands. And these signaling hubs are important for telling each cell type what to do and how they need to function. So using RNA sequencing at the single cell level, we can then start to build these complex molecular networks, where what we're showing is every row is a molecular interaction, and every column is a cell type interaction. So then we can piece together what types of cells are expressing what type of molecular signatures, and specifically understand the signaling that is occurring between those two cell types. And is there specificity to specific brain regions, to specific cell types? And by doing so, we can be able to uncover the specificity for either drug design or gene therapies in the cases of neurological disorder, or even more generally, we can understand the principles of how these two different cell types interact with each other during embryonic and postnatal development to be able to produce a normal, healthy brain. So ultimately, our discoveries uncover truths about how the healthy brain is built, and hopefully identify ways in which we can intervene to fix a damaged or diseased brain. So I'll end with that. So this picture is on the left there is from Cambridge over the Charles River looking at Harvard. Not quite as beautiful as maybe Hawaii, especially throughout the whole year. Cambridge can be quite
kind
cold.
Of beauty.
Yeah. My postdoctoral mentor is up there in the upper right, Paula Arlota, who's given me this wonderful opportunity to study in her lab, and the whole lab is presented down there at the bottom right.
Awesome. Thanks, Jeff. That was perfect. And incredibly interesting because, this is obviously very different from my area of research. But I've, encountered, had friends and colleagues over the years that have studied some of these issues. You mentioned Alzheimer's, for example, and actually our incoming university president, our new university president that's starting just in a month or two is Dr. Keone Kawe, who studies the genomics of the genetic basis of Alzheimer's. And their approach, it's really interesting to see the contrast between what you're doing at this incredibly detailed level, looking at, these specific cell types and looking, contrasting normal and diseased cells by looking at, sort of the RNA products that they're producing, but at this really fine scale. You contrast that with, kind of these genome-wide association studies that are common when looking for things like, genetic, underpinnings of Alzheimer's and whatnot. And so it's really cool to think about, kind of these two, coming at it almost from two different ends of the spectrum, but all with kind of the same goal of identifying, what's actually going wrong, where is it going wrong, and how can we potentially fix it?
Right. Yeah. So I actually use those big genome-wide association studies all the time. Those are coming out more and more frequently now. So for Alzheimer's, for example, it's been known for a few years now that ApoE is one of the, the genes that are driving Alzheimer's disease. What we're understanding now by combining that power with the single cell techniques that we have available to us, that the ApoE levels, the actual gene products themselves, are changing cell type to cell type throughout development and during disease.
Yeah.
So then we can target, a specific cell type. Is it the astrocyte? Is it the microglia? Is it the neuron? And depending on where it's expressed, we need to tailor the design of the drug or the design of the therapy accordingly.
Yeah, cool. So it's really like it's kind of this mutualistic, it's kind of this feedback, right? I mean, you've got
Absolutely.
these kind of genome-wide studies that are kind of, putting the spotlight on a particular gene region, and then you can go in and actually figure out what's, developmentally what's happening and how that can be altered.
Right. And so this is why things get published, so that it can be disseminated throughout the world. It's why we go to conferences, to be able to share all that data so that every lab has access to it and can build off of that knowledge base.
Yeah. Awesome. Well, that was really fascinating. And thank you for sharing all of that. Do you have any sort of parting, any parting words of wisdom that you'd like to share with our budding scientists here at BYU Hawaii?
Sure. Yeah. So science is great. If it's a passion of yours, follow it. There is a place for everyone in science. It doesn't matter what your background is. It doesn't matter what your interest is. You can find a home, and you can make a great career out of it. I'm still in those early phases trying to determine what's next. Obviously, this COVID time is kind of throwing a wrench into some plans, but it opens up other doors. Absolutely. So as long as you're interested in it, you can find a home.
Cool. Good advice. Well, with that, we'll go ahead and wrap this up. And thank you again to Dr. Stogsdill for joining us. Thanks for being here.
Yeah, thanks for having me.